Hello everyone, today I'm going to talk about a crucial topic in public health, Vibrio cholerae, a bacteria responsible for cholera, a severe diarrheal disease that has caused multiple pandemics throughout history. The seventh cholera pandemic began in 1961 in Indonesia and spread rapidly, including Latin America. In Peru, the outbreak that occurred between 1991 and 1998 was devastating, resulting in over 5,000 deaths. This context highlights the importance of studying the strains of Vibrio cholerae that circulated in our region to understand their genomic characteristics and patterns of antibiotic resistance. The objective of this study was to describe these genomic characteristics, the antimicrobial resistance profile, and the phylogeny of the Vibrio cholerae strains isolated in Peru during this outbreak. For this purpose, we analyzed 17 clinical strains collected between 1991 and 1998. The methodology that we used: first, the 70 clinical strains were reactivated and confirmed using the microbiological assays like a TCBS agar and the chromogenic agar. To confirm that they belong to the genus Vibrio, we perform conventional biochemical tests and then multiplex PCR to detect specific virulence genes, which include BCMG, which codes for a specific Vibrio cholerae fragment in the 16S and the 23S rDNA regions. The CTX gene, which encodes the A subunit of cholera toxin, and the TC gene, which encodes the effector protein, a subunit of phyli, specific to the L-Tor biotype. These genes, along with the toxic cooperative agent Phylus TCP, located in the Vibrio pathogenesis islands, are essential for colonization. After confirming this strain that they belong to the Vibrio genus, we perform the whole genome sequencing using the MySeq platform, Illumina. We evaluate the quality of this sequence of the reads using the FastQC program and remove the low-quality reads with Trimomatic. Finally, we assemble the sequence de novo using A5 MySeq and identify the sequence type following the multi-locus sequences type scheme. For phylogenetic analysis, we use RAGS-ML to construct the phylogenetic tree based on core genome, single nucleotide polyform-based, and using the maximal likelihood method. All Peruvian strains grouped as the sequence type 69, which is a well-known and widely studied sequence type. Also, we observed that these Peruvian strains were clustered together in the phylogenetic tree, suggesting the genetic consistency among them during the outbreak. In contrast, the Brazilian strains tend together to be clustered, but there were isolation, indicating a high level of clonal diversity based on the year, as we see that all the strains that were together was in 1995. Also, we identified the CATB-A gene in all Peruvian strains. This gene confers resistance to chloramphenicol, which is an antibiotic commonly used for treating salmonella infections. In conclusion, our findings provide valuable insights into the dynamic of antimicrobial resistance in Vibrio cholerae. We emphasize the importance of continuing to explore the genomic characteristics of these strains Given the widespread distribution of Vibrio cholerae and the amount of the data available in public databases. For future studies, it will be beneficial to include a broader diversity of genomes, which will allow us to gain deeper knowledge about the genetic relationship and evolution of this pathogen in different regions. So thank you for your attention, and if there are any questions, I will be grateful to respond.